All right, so this is the last of the 3 lectures on where we're just reviewing basic genetic terms. And so we've talked about the ones that are single locus, single allele type forms. And we kind of now want to talk about ones that are a little bit more complicated than that, right? So let's see if I can get myself shared here. I tend to seem to always have trouble with that. Maybe I'll learn after 20 years how to do this. Okay, now we should have a screen. All right, so we're talking about modes of inheritance. Again, we've looked at dominant, recessive, codominant, incompletely dominant, lethals, sex-related, both linked and influenced and limited. And I want to finish up with 2 more. The last 2 that was one is the idea of epistatic. Okay, so all these that are kind of lightened out here, we've talked about ones where we're basically really talking about a single locus and the 2 alleles at that locus and how they interact, right? And whether they're on a sex chromosome or not on a sex chromosome. But in reality, all traits are formed by a combination of loci working together, right? You don't get a beard or a brain or an eye or whatever from a single. And how those interact is really what we're going to talk about a lot this semester in forms. One of the terms that's used for that is this idea of epistatic. Okay, this is where multiple genes, usually forms, come together and interact with each other through pathways. Okay, things like Alzheimer's and diabetes and autism are all thought to have an epistatic basis. So as part of the way we try to teach this is we use the simplest form we can. In general genetics, anybody does. It teaches it. We talk about, okay, and really you got biochemical pathways, you got some precursor or some intermediate molecule. There's a gene that puts in an enzyme or puts in a product and whatever it's going to do somehow in that pathway to cause a modification of that precursor to form another product. That product is then impacted by yet another product, and you end up with a final product, right? And so depending on what A does, makes this intermediate product some form that B is going to work on. If A doesn't do its job, B can't do a job here, and you end up with something. If A overdoes its job, you end up with something else. So the point is you've got multiple genes interacting. And then of course we give examples of how that works, right? We give examples for how you take a basic 9-3-3-1, because you're talking about, we always use a 2 locus system, right? And say, okay, how do you adjust that to get to 9 to 7? Well, big A and big B, doesn't matter what, you get 2 of those together, it doesn't matter what you are, you're purple. If you get 1 big A or 1 big B or something else, or 2 little B's rather, 2 little A's, then you're green, okay, or something. And there's not the classic ratios that you spend some time hopefully learning, not memorizing, but 9 to 7 and 15 to 1 and 9-6 to 1 and 12-3-1, etc. 9-3-4, all classic forms of a 2 locus epistatic system, and they're called all sorts of different things. Unfortunately, every textbook and every place calls them something different. We generally call them single dominant and double dominant and double recessive and that. But here, look, they're duplicate and dominant and recessive and dominant. That's one of the struggles that we have, is that every textbook is different. And students will get on the web and they'll come back and go, well, you called it a double dominant, but here they're calling it a recessive, blah, blah, blah. And okay, well, I'm sorry. That's, it's unfortunate, but I don't know of any standardization. What's standardized, of course, are the ratios, and you can call them what you want. And that's the way we teach it. And certainly that's a good example of how it works. But reality is that, when we start talking about this, we're really talking about multiple interacting pathways, and it's not just epistatic at a single locus, or single pair of loci. We have these pathways. This is supposed to be a cell. There's supposed to be some nucleus in here. And you end up with these multiple proteins involved, going in and signaling all sorts of different ways for different DNA to be produced, and how that DNA interacts with other DNA in the mitochondria or wherever it's going to go. And then those come back and interact over here. Those are all epistatic interactions. In fact, you get these multiple interactive pathways to produce what is truly a cellular phenotype. So yeah, this this little pattern over here definitely explains it and you understand it. In the real world, when we say epistasis, we really mean that, lots of genes will be involved, right? In fact, this is a pathway here which is dealt with. Somebody did an experiment where they changed diet. They looked at males and females, and these are the epistatic pathways that are all interacting with each other and these little outside show you the genes that are supposed to be involved on which chromosomes they were located on and which genes then responded in various ways and cross patterned back to others and that's formed. So point is it's an important concept and it's going to be important for us because we're going to talk about things. A lot of genetic diseases, things are epistatic. Okay, it's because you have a mutation in one locus affects this whole suite of other loci as they respond either from the cellular function or as from a pathway function. Okay, now the next thing and the last thing of all this is, when you look at sequence data, what do you see, right? Gosh, well, this is just sequence data. So this is some sequence we've lined up here at the top. And then all these others are variants that were found in some other subsequences that were made. And this could be, a whole family or a tribe or a group of people or just a bunch of, or tissue from different organs or whatever. But the point is you get lots of variation. All these spots that you see in here that aren't dots are variants, right? This is one gene. It's not even a full gene. It's just 700 bases of a gene region. Doesn't matter what the gene is, but it's there. And you see everything. You see indels. You see things where you've got deletions, or here in this case, insertions. Here you've got deletions. You've got all these different variant forms. Every one of those is an allele, right? Every one of those, it's different. If you take this one and compare it to that one, that's an allelic form of that. That's an allelic form of that. So you get these multiple allelic forms, all there. Some of them have absolutely no effect on anything, right? They're silent. It doesn't matter if you're a G or an A right there, big deal. Doesn't matter if you're a G right there. Others, that one makes a completely dysfunctional protein or something, right? And so it changes the allelic expression. So, in the real world, we think about two alleles and, oh, by the way, we got a locus and, and here you've got big A and you got little a. We don't. You have big A prime, big A triple prime, little a triple prime. Oh, by the way, it's AC, AG, AT, AB, all of that going on, okay? So that's one of the really big things that we're talking about here. We're talking about the fact that in the real world, we have multiple alleles. Now, take all of that, and you say, okay, almost every trait we think about that's important to us, right? Developmental traits, language, vision, thought process, body morph, whatever. Those are polygenic, right? They're polygenic. There's lots of genes. So those pathways all intersect and they work to function. And this gene that works in this pathway over here may contribute to a totally different pathway over here. They're polygenic. Things like IQ, weight, cancers, all those are polygenic. They all have multiple allelic forms, okay? And they also all have an environmental input, right? There's almost no gene that's a simple dominant recessive that, irrespective of what the environment did to it, it doesn't matter. And that can be an external environment, heat, temperature, humidity, whatever, or it can be internal cellular environment being different. So, they all play a big role. And certainly the traits, a lot of things we're going to talk about and think about will have these massive pathways that we've already looked at when we talked about cell structure back there. And we'll have multiple allelic forms. So something you have to remember. Okay. Well, I've covered a lot of these forms, a lot of these different types. I think that's enough for you. But the point was, again, just so in your head you went, oh, codominant. Okay, what's that different than a sex-linked? Okay, I got that, right? Now remember that you can be sex-linked and codominant at the same time, right? And so these all fit together. But basic idea is just so you read a paper, or when I spit something out about something being recessive lethal, your brain has at least a concept of what it is. Okay? All right, that concludes that, and we will now sit down and think about the next lectures, which should be, I think, probably developmental. All right.